So thank you so much to the ISHR for the opportunity today to speak. I really appreciate it. All right, so in the heart, there are several cell types, the contractile cardiomyocyte, as well as the interstitial cardiac fibroblast, which is the cell type I'm going to be focusing on today. And in the healthy heart, the cardiomyocyte provides a contractile function, while cardiac fibroblasts have multiple functions, including matrix deposition and degradation, as well as induction and electrical coupling. But in the diseased heart, in states such as hypertension, the cardiac fibroblast can become hypertrophied, leading to heart wall thickening, and the cardiac fibroblast can become activated into a form that's commonly referred to as the myofibroblast. And in this form, it secretes extra ECM, leading to cardiac fibrosis. And the combined effects of hypertrophy and fibrosis can lead to heart failure. And nearly 6 million Americans suffer from heart failure. And because cardiac fibrosis is so complex and difficult to treat, this is one main contributor to the fact that cardiovascular disease is a leading cause of death in the US. And so cardiac remodeling can be triggered by a couple different things. One being a more acute stimulus, such as myocardial infarction, or a more long-term stimulus like hypertension, as well as aging. Our lab is interested in hypertension, and this figure does a nice job of showing that trigger phase where the fibroblasts are activated. And then when the stimulus persists, such as in hypertension, we move into a chronic phase where the activated fibroblasts, there's crosstalk between them and immune cells, as well as the cardiomyocytes, and many feed-forward mechanisms involving fibrotic and inflammatory signals that lead to ECM deposition. And so in hypertension, there are elevated levels of angiotensin II, a vasoactive peptide, that can increase reactive oxygen species generation through a variety of mechanisms. And these reactive oxygen species can lead to elevated chemokines like MCP1, and therefore increase immune cell infiltration into the heart and exacerbated inflammation. And although the fibroblasts can react to the inflammation, I want to point out that they can also actively secrete inflammatory signals as well. In addition to this, angiotensin II upregulates TGF-beta, which activates a number of signaling pathways that lead to fibroblast proliferation and transdifferentiation into that myofibroblast form, leading to increased matrix deposition in the heart. And so the study that I'm presenting today uses single-cell RNA sequencing. And so for those of you that are unfamiliar with this technique, it's a high throughput method for gene expression in individual cells. And using a microfluidic device, individual cells are encapsulated in oil droplets that contain the reagents for reverse transcription, as well as a unique DNA barcode. And so after PCR, all of the cDNAs for that individual cell are tagged with that unique barcode. And the aim of this study was to identify cardiac fibroblast subpopulations by differential gene expression in cells isolated from the hypertensive rat heart. We utilized adult male spontaneously hypertensive rats. And what I'm presenting today is just one part of a larger study where we used control SHRs, as well as a group that were transiently treated with an ACE inhibitor. For those of you that want to hear the interesting effects of transient ACE inhibition, please tune in on Friday to hear Dr. Tabin Hale speak on that. However, today I'm going to focus on the cells coming from the control hypertensive hearts. We isolated the fibroblasts from the left ventricle from a total of 7 animals and subjected them to single-cell RNA-seq, again looking at differential gene expression as well as pathway analysis using the David functional annotation tool. Downstream analysis by UMAP identified 9 clusters, and if you're not used to looking at these sorts of plots, I just want to point out that each individual dot here is an individual cell, and how close together they are implies how similar they are to each other in terms of gene expression. And these 9 clusters are labeled as 0 through 8. And when we look at the proportion of cells that distribute into each of these clusters, 7 and 8 collectively only contribute less than 2% of the total fibroblast population. So today I'm going to focus on clusters 0 through 6. And the next step was to determine how these clusters differed phenotypically. And so we did differential gene expression to compare one cluster compared to all others. And we focused in on processes that of proliferation, inflammation, and fibrosis, as these are key factors for fibroblast expansion and activation. So all I want you to take home from this large heat map here are that these are select genes that were differentially expressed in these clusters in the processes of interest. And I want you to see that in clusters 0 through 3, there are more extreme characteristics, more highly fold changes between clusters than there are in 4 through 6. So I'm going to focus on these 4 clusters for the remainder of the talk. In cluster 1, when compared to all others, genes for proliferation are significantly downregulated, while those for oxidative stress, including these chemokines, and fibrotic genes, including markers of fibroblast activation like alpha smooth muscle actin and FAT5 and CTGF, are all significantly upregulated in cluster 1. However, in cluster 2, when compared to all others, we're looking at almost the complete opposite situation where genes for proliferation are significantly increased, while chemokines and genes related to fibrosis are significantly decreased in cluster 2. When looking at cluster 3, there were no unique characteristics for proliferation. However, chemokines were reduced, but there was a mixture of genes related to fibrosis being either up or downregulated here. I do want to point out that CCN3 and CCN5 are genes that have been shown to actually suppress fibrosis as well. And lastly, in cluster 0, we're seeing main characteristics of significant reductions in chemokines and genes related to fibrosis. And so this is an overall look of genes in these areas that were significantly characterizing these clusters. And so when we did the pathway analysis for our data set, we found that in cluster 1, pathways related to wound healing, inflammation, collagen biosynthesis, and stress fiber assembly were significantly upregulated. And so we dubbed this cluster our activated fibroblasts. For cluster 2, pathways related to cell division are significantly enriched, while those for fibrosis, including collagen biosynthesis, are significantly decreased. And so we call this our proliferative cluster of fibroblasts. When we look at cluster 3, similar to that set of fibrotic genes that had a mixture of increased and decreased expression, we see the same in our pathway analysis. So fibrotic pathways such as TGF-beta, stimulus, and cell adhesion, as well as response to wounding and inflammation response, are both increased and decreased. And so this is a moderately fibrogenic cluster of cells. And lastly, in cluster 0, we see a significant downregulation in fibrotic pathways like wound healing and collagen biosynthesis. So this is our least fibrogenic cluster out of the clusters that we've identified. And so lastly, we did a pseudotime trajectory analysis where all of the cells are plotted in time to see how they may relate to each other in time. And interestingly, so this is for the whole data set, both control and ACE inhibitor treated. When we color this map for treatment, interestingly, the majority of the cells to the left of this line are from our ACE inhibitor treated animals that Tabin will speak on Friday. So I'm going to focus on the cells on the right side, which are mainly from our control animals. And you can see all of the branching and the lines that were generated from the pseudotime analysis, and this was unsupervised. So based on the gene expression, we're looking at cluster 2 here as potentially being an expanding population of fibroblasts that then lead into one of 3 potential cell fates. One being cluster 3 here, that moderately fibrogenic cluster. Another being cluster 6, which you may remember wasn't super remarkable in terms of fibrotic or inflammatory genes, but because it looks like a gateway into that activated cluster 1, it may be of high interest because of that. And lastly, clusters 4 and 5 form somewhat of a bridge to the left side of this plot that's populated by mostly treated cells, and also an expanded cluster 0, which was our least fibrogenic cluster. And so I think that this is an interesting layout of how these cells may relate to one another. And so in conclusion, the hypertensive left ventricle contains 9 distinct subsets of fibroblasts that are characterized by different phenotypes, including proliferation, fibrosis, and inflammation. And the contributions of these subpopulations to wound healing may help us get a better understanding of the complex signaling that occurs in fibrosis and therefore may help identify potential therapeutic targets given subpopulation location in the left ventricle, as well as timing of their expansion in different disease states. And so with that, I would like to thank my lab, Dr. Tabin Hale and my coworkers, as well as our collaborators at TGen here in Arizona, Dr. Huntelman, Salboon, and Matt DeBoth, who is our bioinformatician, and our funding sources. So thank you very much for listening.
So the first question is for BckA from Gary Loposchak. Nice talk. How do you think BCAA are modifying phosphorylation of AKT? And then the follow-up question, are they interacting directly with AKT?
Thank you, Gary, for the question. We did see that BCAAs could directly activate mTOR signaling. So we think that that is one of the way that it is modifying AKT phosphorylation. And also we found that BCAAs can inhibit insulin-induced PP2A inhibition. So that is also another method that we feel. And as it is known, and we have also found that BCAAs can directly impact mitochondrial respiration and increase ROS, so which can also influence AKT phosphorylation. So right now, actually, we have a couple of theories surrounding this.
The next question we have is from an anonymous attendee. They're talking about the fibroblast cell cultures. They ask, is cluster 8 a non-fibroblast cell culture the most deviating one, is what they mention. So I believe this question is for Alexandra Garvin.
Yes, thank you for that question. We were worried about the same thing when we first saw the clusters. However, none of our cells express markers of either endothelial cells or smooth muscle cells or even hematopoietic cells. And so we know that these are fibroblasts. However, they are just a very unique subset, as is cluster 7. And one theory we have is that potentially these fibroblasts may potentially expand in different either injury states or other ways, but we're certain that those are fibroblasts. Why they're so unique, we're not incredibly sure.
Okay, so the next question is from Julie. It's a technical question for BckA. How did you measure BCAA level, whether you use a mass spectroscopy?
Yes, we indeed used UPLC MSMS for the intracellular as well as the serum and plasma BCA and BCAA levels, yeah.
Then we're going to jump back to Alexandra. The next question is, did you do spatial selection of the fibroblasts you subjected to RNA sequencing?
I'm sorry, I'm not exactly sure what you mean by spatial selection. If you mean localization to the heart, we just did a complete LV digestion and isolated all fibroblasts from the left ventricle. I'm sorry if that doesn't answer your question, but.
Okay, so the next question is also for you, Alessandra, from Sina. Is it possible to distinguish which portion of the heart each cluster of fibroblast comes from, that is closer to the epicardium versus closer to the endocardium?
As of right now, our analysis does not distinguish that, but some follow-up studies that we will be doing is some imaging in the heart and staining with some markers of these clusters in the left ventricle to see if potentially there are special localization for some of these clusters. And I'm very interested in that, actually, to know if some of these phenotypes are coming from different areas of the heart. So hopefully that is to come, but right now we don't know.
The next question is also for you, Alexandra. They ask, did you compare these fibroblast clusters to a non-hypertensive rat ventricle fibroblasts? And if so, which clusters are specific for hypertension?
We did not compare these to non-hypertensive hearts. The main question for the study was within the hypertensive heart, what the impact of ACE inhibition was. And so we did not have that non-hypertensive group in this study, and we haven't done that.
Okay, so the next question is also for Alessandra from Leslie Kennedy. So I think it's similar question what you just answered, but still I will just read out. Is there any evidence of fibroblast in differing region of ventricular myocardia having distinct gene expression? In other words, do you think the cluster group in cardiac region, or do you think the clusters may be more heterogeneous, spread throughout the myocardium? This may impact crosstalk between fibroblasts and cardiomyocytes in the left ventricle versus those are in the right ventricle.
Yes, great question. And I agree with you that potentially different localization of these clusters would impact the crosstalk. And although right now we don't know, I wouldn't be surprised if we find some of these clusters are in particular areas. I know that there's some evidence out there to suggest that fibroblasts from either the endo or the epicardium serve slightly different roles. And I'm eager to know the question to this, or the answer to this question.
Great. The next question is, how do you isolate the LV fibroblasts for single cell RNA sequencing?
Sure. So after we isolate the left ventricle, we mince up the tissue and put it through a couple rounds of digestion with an enzyme called liberase. And the fibroblasts that are released from that digestion are then plated, and since fibroblasts are so adherent, we use those adherent collagen producing cells, which is what we used for this experiment.
Alexandra, may I just interject? So maybe they mean, which methodology do you use to isolate cells? Do you use Langendorf, non-Langendorf methodology?
Oh, so we don't cannulate the hearts, and we don't digest the heart when it's in its full form. I know some people will perfuse an enzyme, but what we do is we just mince up the tissue, after it's removed, and then from that minced up tissue, we submerge it in the liberase buffer and put it through several digestions and collect the supernatant, and then let the cells adhere, if that clarifies.
Okay, so next question is from Ron. He's asking two questions. Were the RNA sequence experiments from the hearts at a time point with over tissue fibrosis? And the next question is, any plans to build a progressive data set over the progression from hypertension to HT to dysfunction?
Yes. So at this point, the hearts are certainly hypertrophied, and there is an amount of fibrosis, but it's not overt. I would not call it overt fibrosis. If you follow up with other injurious stimuli, you can stimulate higher amounts of fibrosis in these hearts at this time. And as for the second question, any plans to build a progressive data set? We would love to with RNA-seq, and it is, I think, an overall goal to understand the timeline of these changes, but right now we're focusing on how responsive this cell type is to our treatment that you'll hear about on Friday.
Oh, sorry, I'm talking when I'm muted. The next question's for you, Alexandra. From Mohan, they ask, could you please elaborate on the enzyme cocktail used for single cell preparation?
I think I already answered that. If you mean for cell isolation, we use liberase as our enzyme.
Okay, so the next question is also for you, Alessandra. This is from Jun Sun. He's asking, besides co-treatment with ACE inhibitor enalapril in angiotensin II induced hypertension, have you tried to treat the rats with nitric oxide donor? If so, is there any difference in cell cluster between angiotensin II plus enalapril and angiotensin II plus NO donor?
So good question. We haven't done those exact experiments, but previous experiments in our lab have utilized a NOS inhibitor. So not a donor, but in our hypertensive hearts that either got the transient ACE inhibition or not. They also then followed up with an L-name NOS inhibitor. And in those studies, isolating the cardiac fibroblasts and then looking at them in vitro, our transient treatment does protect against some of the impacts of that L-name treatment in terms of proliferation and collagen gene expression. So we know that this transient treatment does play a role in protecting against some of those pathways, but in terms of the single cell sequencing, we haven't done those experiments.
All right, the next question, and maybe if individuals would like further or additional information, they can feel free to reach out to the individual speakers if they feel like the answers weren't adequate to kind of what they're asking for. But the next question to Alexandra as well is, while classifying the 9 subpopulations into 3 classes, what were the criteria being used to group them into 3? For instance, does it mean the subpopulations of fibroblasts classified under oxidative stress are programmed to express different kinds of oxidative stress response genes?
So the way that I went about, going through this data is we have thousands of different differentially expressed genes, and what we were looking for were, I chose these 3 categories because of the roles that they play in fibroblast activation. And so in terms of actually grouping a cluster into one of those areas, that's a little bit harder to do, but more just looking at overall, was this cluster more proliferative or more inflammatory or more fibrotic, was a way for us to help understand potential contributions of these clusters to overall, cardiac fibrosis. And so I wouldn't say that we've necessarily pinned them down to be one or the other, but just over all of the genes that were, uniquely expressed in a cluster, we pulled out these processes because they were relevant to our questions and because they were areas where we saw some unique characteristics between the clusters, just to help understand their contributions.
So Alexandra, Richard is wondering, what are the markers did you use to identify your fibroblast population after isolation?
Markers in terms of purity, I think, is what they're asking. So ultimately, we know that all of the cells that we isolated express things like vimentin, and that they don't express markers of other cell types that we may potentially get in this prep, such as vascular smooth muscle cells or endothelial cells or immune cells. We didn't see CD68 or CD31, smooth-lin, any of those markers of potential contaminating cells in a fibroblast prep. So we're confident that we're looking at mainly, collagen-producing fibroblast cells.
All right, the next question from George is, do you know if the change in fibroblast cluster morphology in response to hypertension is terminally differentiated or reversible? And then there's a second question. Well, I'll read them both, then you can answer them both. And the second question is, do you know whether female mice undergo the same transformation in fibroblast differentiation in respect to insulin, given the sex-specific differences in collagen deposition?
Okay, great questions. So the first one, we don't know in terms of, differentiation, whether these cell, states may be reversed either with a treatment or, if they are reversible at all, or if potentially these cells might, die out and be repopulated. We don't know if these are reversible phenotypes. But all interesting questions in how, a targeted treatment may impact these clusters. And then the second question, we have not done any studies with females, but of course that would be in the future directions, and knowing how different they are and how relatively protected a female heart is, it would be interesting to see if these same clusters would come up.
Okay, so now Monica is asking how I missed it. Okay, so pure, how you isolate pure population of cardiac fibroblast from the heart. Then the second part of her question is also, it seems that you have 4 distinct population of fibroblast. Wouldn't you expect to have more visible transition from one population to another?
So I think I've, hopefully you've heard my answer to how we isolate the fibroblasts. So I'll, in the interest of time, move on to the second part. So we get 9 distinct clusters. I focused in on 4 clusters today, given their unique characteristics in areas of proliferation and fibrosis and inflammation. And so whether I would expect to see more visible transition from one population to another, if you're referring to the UMAP plot, where it's basically a cloud of cells with varying colors, I think that has to do with the fact that we're using a very pure population here. They were, rested in, culture dishes for 3 days. So they were adherent. They were growing for 3 days. This is a very pure population. So we don't get huge changes in gene expression, like you might even see with different cell types in the heart when people isolate all the cells from a heart. So because these are, cardiac fibroblasts that were cultured for 3 days and then subjected to single cell RNA-seq, the fact that we even saw distinctions was exciting. And even more so the fact that we see an impact of our treatment, again, which you'll hear more about on Friday, was even more exciting. So I can't say that we would have expected to see a more visible transition given the fact that we were working with such a purified population of cells.
Great, thank you. We're going to switch over to Natasha now. Natasha, the question for you is, would C144S alter the ubiquitin ligase activity of TRIM72?
Okay, thanks actually for that question. So that was one of the first things that I actually looked at in hearts from these mice, because its ubiquitin ligase activity is believed to affect IRS1, cause its degradation. So I tried to look at overall ubiquitination in the hearts, and I didn't see a change in that, and I didn't see a change in targets of TRIM72 that are expected to be changed if its activity was changed. Having said that, I did not specifically run an assay looking at TRIM72's ubiquitin ligase activity. So if that answers the question.
So the next attendees is asking or wondering whether the effect you see is the enzymatic dependent protection or structural.
So sorry, I'm not specifically sure what that means, but what I'll say is I didn't see evidence of enzymatically affecting its enzymatic activity in terms of ubiquitin ligase activity. But other than that, I don't know the answer to that question.
All right, we're going to switch back over to Alexandra, and I believe you've already asked or answered this question, but just to make sure it's adequately addressed. The question is, did you find any differences in the cluster depending on the location in the heart?
Right, and unfortunately we don't know. We isolated fibroblasts from the heart altogether, and so unfortunately we don't know where each of these clusters are coming from, if they are specifically located, but something we plan to follow up on with some candidate markers using some imaging of the left ventricle.
Let's go back to our first speaker, BckA. So Azrul is asking, what is the glycogen content? The second question is, does it affect Krebs cycle intermediates?
Thank you for this question. We did not measure the glycogen content, but there has been studies, a recent study actually in a PPM1K phosphatase knockout mice, where they did find differences in glycogen metabolic intermediates, and they also incubated HepG2 cells, liver cells, with BCKAs, and they found an induction in glycogen phosphorylase mRNA level, which is responsible for glycogen breakdown. But it will be interesting to see what happens within the cardiomyocytes. For the second question, we are in the process of developing a method to measure the TCA cycle intermediates. But then again, previous studies have shown that if the BCAA oxidation is inhibited, that results in reduced TCA cycle intermediate. So we are expecting a similar result with our study as well.
Thank you. All right, we're going to come back to Natasha. Was there a sex difference in the ischemia reperfusion results, and are females also cardioprotected?
So we didn't specifically compare male versus female TRIM72 knock-in mice in the ischemia reperfusion experiments. But having said that, we had a couple of female mice that were done, and they also showed some protection compared to the wild type mice.
So the next question is from Emma, and it's for Alexandra. I think the Srinivas part is you already answered, so I'm directly going to the second part of her question is, have you compared your single cell data and cluster with other model, such as the more ischemic model by BckA et al. 2018?
Yeah, thanks for that question. And I know that paper you are speaking of, and if I recall, they looked in all cell types of the heart, cardiomyocytes and fibroblasts, and that they did find very distinct clusters. They found fewer clusters than we did, but very distinct ones. And what I have found in general is when comparing the clusters that we have to those from models of ischemia, post-MI, that those clusters tend to have much more extreme expression of markers such as periostin and these other markers that are really highly upregulated after an injury that we're not necessarily seeing differentially expressed. They're expressed in our cells, but not very highly, and not in distinctive clusters. And so in terms of comparing our data sets, I think it's relatively difficult given the methodological differences, but also I think ischemic hearts might show more extreme expression of some of those markers that we're not necessarily getting in our hypertrophied heart. I hope that answers your question.
The next question's from Svem and Alexandria. They're wondering if you compared your hypertrophy data set with the single cell data from mice after MI published by Richard Harvey's lab. And is there a specific cluster in both data sets contributing to cardiac fibrosis and scarring?
So thanks for that. And I think my answer will be similar to my previous one in that I can't say I've compared it to the specific data set, but to MI data sets in general, those data sets tend to have more extreme expression of activated cell markers. But whether there's a specific cluster in the data sets contributing to cardiac fibrosis, I can't speak for the other data set, but for ours, our cluster 1 really tends to express genes that characterize, an activated phenotype. And so in our data set, we're really zeroing in on really one cluster being the most activated and potentially most pathogenic. And so it'll be interesting to see how, our further verification of these clusters and potential localization in the heart, how this might impact overall cardiac remodeling.
Okay, so a couple of technical questions for you, Alexandra. So when fibroblasts were plated, what substrate did you use? And the second question is, and how long were the fibroblasts were plated?
Yes, so we plated on plastic, and they were plated for 3 days. And we are well aware of the activating that plastic can activate the fibroblasts. And so knowing this, you know, as we look at our data set, it's almost interesting to see, especially with the impact of our treatment, the fact that, so we recognize this as a potential limitation, but also the fact that plastic can potentially activate our cells. It may also help pull out the differences between our treated and control cells as well. So yes, we plated them for 3 days. They did not grow to complete confluence, but they were plated on plastic, just to slightly expand the population for the sequencing.
Thank you. We're going to jump back over to BckA. What effect does BCAA have on the respiration capacity of the cardiomyocytes?
We measured the effect of BCKAs on the respiration capacity by seahorse, and we did find that it suppresses the basal as well as FCCP-induced maximal respiration rates. And it also resulted in reduced ATP production within the cardiomyocytes.
Okay, so the next question is for me. So thank you, June. This is ECI ISHR banner, but no, it's not from the big TV screen. Due to intellectual property, I cannot disclose the exact mechanism. But jokes apart, next question is for Natasha actually. And so the question is TRIM72 specifically expressed in cardiomyocyte or other heart cell as well? How do you think, that second part of the question is, how do you think the nitrosylation at cysteine 144 affect the other cells such as fibroblast? Third question, does cysteine 144 nitrosylation affect ROS generation?
Okay, so first, TRIM72 has been reported by some recent papers to affect fibroblasts, so it seems to be expressed in cardiac fibroblasts. I don't know how nitrosylation cysteine 144 would specifically affect fibroblasts. What I can say is that there was a cell culture experiment that was published in Mark Kor's paper a few years ago before this study was done. And it also showed that this specific type of mutation, where you get cysteine 144 mutated to serine, protected against hydrogen peroxide-induced cell death. And so, I mean, I would expect that as nitrosylation would also protect against other types of cell death. And then as far as the effect on ROS generation, I don't know. I didn't look at that, but I would expect that by protecting against further injury, you would keep there from being as much ROS generation, but I didn't test that. Okay.
Great, and the last question for you, Natasha, from Jader is the specific mutation in TRIM72 affecting mitochondrial function during ischemia reperfusion injury?
Yeah, I would love to know the answer to that question, but I did not look at that during ischemia reperfusion injury. And again, I would kind of expect that by protecting against further injury, the mitochondria would be overall healthier compared to the wild type mice, but I don't know that that would be a specific function of the TRIM72 mutation versus a byproduct of the protection.
Thank you, everyone. We've reached the end of our questions, and so I think we'll go ahead and wrap up at this time. We just want to thank one last time the 3 presenters that we had today. Thank you so much for participating in this webinar and for sharing your science with us. We really appreciate you being here today. And one more plug for the other webinars that we have ongoing this week. Please check them out. We have some great science that's going on here. So thank you for joining us today.
Thank you.
Thank you. I mean, if you don't mind, I would just like to personally thank the three of you. Dipshika, Alexandra, Natasha, great stuff, great webinars. You were a bit of a kind of, to some extent, guinea pigs, because this was the ECI-MCI first webinar, and I think it went really well. We had great numbers of attendees, and I think the Q&A session was fantastic. And last, I'd like to thank Sam and Sarah. Great sharing. I mean, they've constructed this, by the way, this ECI-MCI session, and I'd like to thank them because I think this is really needed, as on top of, obviously, you know, the other webinars that we've had. And we've got 2 more coming up as well. Is that right?
Yes,
Yeah.
there are 2 more. Yeah.
Yep.
So please come and join us, everyone. So thank you once again.
Thanks, guys. Have a wonderful day.